Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about polymerase chain reaction. So polymerase chain reaction is a technique basically carried out in a thermal cycler machine. Thermal cycler machine can produce million copies of DNA in a second, in rapidly. Okay, so using that machine we can produce many copies of DNA very short time, in a very short time. So this process is called as polymerase chain reaction. Okay, so basically the whole process is divided into three phases. The first phase is denaturation, second phase is annealing, and the third phase is elongation. So first denaturation, where the DNA gets denatured or double stranded DNA gets separated from each other and single stranded DNA is produced. Then primer annealing. In the process of primer annealing, two primers attached to each end of two DNA strands. Then extension, primer extension, that means DNA polymerase. By the help of DNA polymerase, the polymerization process was carried out to make a copy of existing single stranded DNA. So in this way, two copies of each strand is produced. So this experiment was first discovered by Gary Mullis in the year 1983. So in 1983, Gary Mullis first discovered the process of polymerase chain reaction, or PCR technique. This technique can be carried out in the machine called as thermal cycler. Thermal cycler, or you can tell it thermocycler. Okay. Next. So PCR is a laboratory technique that amplifies a specific DNA region. So that specific DNA or gene of interest, we have to keep it for a longer time. So we can produce many copies of that specific DNA in a very short time using that machine by the process of polymerase chain reaction. So the instrument used is thermal cycler. This instrument can have plate or block format, okay, with many wells. So Gary Mullis developed PCR in 1980s, so particularly 1983, in the year 1983, and for that he won a Nobel Prize for his work for a decade later. Okay, next. So how it works? PCR uses thermostable DNA polymerase, because the process of PCR is carried out in a very high temperature, because for denaturation above 90 degrees Celsius temperature is used. Okay, so at that temperature only thermostable DNA polymerase can act, and that particular DNA polymerase is called as TAC polymerase, which is specific to this PCR technique called TAC polymerase. This polymerase is extracted from Thermos aquaticus. That Thermos aquaticus is a thermophilic bacteria, or sulfur spring bacteria have this type of polymerase, TAC polymerase. This polymerase is used in this PCR process. So at first denaturation, the reaction mixture is heated at about 94 degrees Celsius to separate the double stranded DNA into single strands. So double stranded DNA separate from each other to form single stranded DNA in the process of denaturation. Next is annealing. The temperature is lowered in the process of annealing. The temperature is about 60 degrees Celsius, 55 to 60 degrees Celsius. So here oligonucleotide primers added at each end. At each end means, suppose to the 3 dash. So the oligonucleotide sequence is added at 3 dash end of both the strands, because it can form DNA in 5 dash to 3 dash direction. Okay, so primers are attached in both the direction. Okay, next step is extension. So Mullis, during his work, he used Kleno fragment of the DNA polymerase. So polymerase 1, the DNA polymerase which is used in this process, have many functions, like it can act as polymerase in 5 dash to 3 dash direction. It has also 3 dash to 5 dash exonuclease activity. Okay, so it can remove the enzyme. Okay, so that is called as Kleno fragment. So he used Kleno fragment during his experiment. Okay, so in this way, by the process of extension, another strand is formed. Another 2 strands will be formed from existing 2 strands. So in this way, we can create many copies by following many cycle of reaction. Okay, so it is the whole process of polymerase chain reaction, which is consist of 3 steps. One is denaturation, second one is primer annealing, third one is elongation, finally producing 2 strand of DNA from existing 2 strands. So basically from 2 strand, by following one cycle, we can produce 4 strands, 2 old strand and 2 newly formed strands. Okay, so in this way we can create many copies, billion to million copies of DNAs in rapidly or very quickly in just 5 to 10 minutes. Okay, so it is very important process in research, in medical field. Okay, so it is all about polymerase chain reaction. I hope you really enjoyed this session and will get benefit from this lecture video. So if you liked it, please hit the like button, please share with your other friends, and please subscribe to my channel Sunanda's tutorial. Thank you.